In this last video, we are going to look at some neuron histology as well as histology of the spinal cord, specifically a cross-section. But first, we need to talk about some of these neural membranes. And these should be familiar looking terms to you. We've seen all of these prefixes before with the muscle and skeletal unit, peri, endo, and epi. In this case, our suffix is neurium, referring to the neural system. So we're going to have a layer of connective tissue surrounding this entire nerve structure, and this is called our epineurium. It's kind of the broadest scale that we talk about. And then we have perineurium, or perineurium. This is going to surround a bundle of nerve fibers. So it's that intermediate membrane. And then basically what we're seeing projecting here is a nerve fascicle, a bundle of nerve fibers or nerve filaments. And so each of these individual nerve filaments is going to have a little connective tissue membrane called the endoneurium. So this should look very similar to that organization of the muscle system and the muscle membranes. What that looks like histologically is, if we zoom out here, come on now, there we go. So histologically, we see the same pattern. This could be a little trickier for students to identify on a slide, so I'm not going to emphasize this on the lab exam, but that outermost layer usually shows up a little lighter stain on slides. This is going to be our epineurium. Perineurium is that intermediate layer, and you can see here it does have a color change when we go from epineurium to perineurium. This is a little darker stained. And then basically each of these tiny tubes you see is a nerve filament. And surrounding each of these nerve filaments is going to be our endoneurium. So good to know, but not something I'm going to emphasize on a lab exam. Okay, let's switch over now to our favorite histology resources. So we're going to talk first about the basic structure of the neuron, which should hopefully be reviewed for you. And then we will look at two specific examples found within the central nervous system. So first off, we have our basic neuron. In this case, this is a motor neuron. And so what every neuron will have is dendrites, or at least one dendrite. These are going to be these branch-like structures. They are receptors for information. So when we have a lot of neurons strung together, axons are going to be sending information away from the neuron, and dendrites are going to be receiving that information to create that communication system. Every neuron is going to have a cell body. So the roundest part here that looks the most like a cell, that is your soma. Within the soma, we would expect to find a nucleus, and in this case, we can't even see the nucleolus, that mini nucleus. Sometimes, if you're lucky, your slide will show an axon. This neuron here does not have an axon that is viewable. So depending on how the slide is stained, those can show up better, worse, in this case, not at all. One important thing to know about neurons is that they're going to form synapses. So that junction of communication between the dendrites and the axons of other neurons is called a synapse. So make sure you're comfortable. The cool thing about this site is you can pull up a slide like this, and then if you want to check your work and make sure you're identifying the correct structures, all we have to do is click Show Labels. So I would use this as a study tool when you aren't able to be in lab. All right, our next neuron is going to be found in the cerebellum. And a lot of the neurons, the specialized neurons that we talk about, are going to be multipolar in shape. And really all that means is they're going to have multiple dendrite projections coming off of that soma. And this is going to allow for a rapid exchange of information. So the more dendrites a neuron has, the easier it is to communicate lots and lots of information. So most of the neurons that we talk about in the CNS are going to be multipolar. So our first example is going to be Purkinje cells, or Purkinje neurons. These are a specialized type of neuron. They are found in our cerebellum, which, as we learn, is going to be associated with things like coordination and balance, so a lot of motor control. So what we see here is a large-scale view of our cerebellum. We're going to talk about what some of these branching structures are, but basically we've got white matter and gray matter. The white matter is actually the kind of inner branching we see here, and then filling in between that we have our gray matter of the cerebellum. Let's zoom in now. So we'll go ahead and click on the Purkinje layer. And I bring up gray versus white matter because that is one way to help us identify our Purkinje layer of neurons. So these neurons are found on the border between our white matter and our gray matter, the inner white matter, outer gray matter. So when you're looking on your slides in lab, I want you to find this border and then zoom in, and we should start to see these guys, Purkinje cells. So let's go ahead and let the app do the work for us of finding these. So this is a Purkinje cell. And you can see it's a little messier than that standard neuron shape we had. What we can see is we've got lots of branching dendrites. So this is a multipolar neuron, enabling that rapid exchange of information. So the main thing to know here is these guys are going to help regulate motor control. And the way to find them is to find this gray versus white matter border and then zoom in on your microscopes. So those are going to be our Purkinje cells. Specialized neuron number 2 is found in the cerebrum. So we've switched over to the cerebral cortex. Kind of looks nondescript here, so we're not going to see too much difference between the white and the gray matter from this perspective. But if we zoom in, you can start to see a little bit of color variation here. So just like with the cerebellum, our gray matter is going to be more external. Our white matter is going to be deeper within the cerebral cortex. And we'll talk about all of that when we get to the brain next lab. But for now, let's click on a pyramidal neuron. So this is that second specialized type. It is called pyramidal because it is shaped like a pyramid or a triangle. And in this case, these neurons are going to be part of the primary motor cortex of the cerebrum. So they're involved with controlling voluntary movement. And because of that, because we have so many complicated motions, a lot of information is going back and forth from the brain to our muscles, yada, yada, yada. These are also going to be a multipolar neuron. So we're going to have lots of dendrites branching out in many different directions. Okay, these two that we talked about, Purkinje and pyramidal, are good PowerPoint questions for the lab exam. So make a note of that. These might show up, or the images from our modules might show up on a PowerPoint slide. Lastly, before we get to the spinal cord, I want to cover microglia and astrocytes. So these guys are not emphasized. I want you to be able to identify glial cells, those support cells, on a slide. And then this is going to be maybe PowerPoint candidate questions, but not something I would expect you to identify under a microscope. So microglia and astrocytes are kind of non-neuronal cells. They're going to help maintain and support our nervous system. So on the left, we have microglia in black. So we can kind of identify them by the dendrites here, branching dendrites. So that's a microglia. This is a microglia. This is, and this is. So we're seeing a lot of branching, and basically these microglia are going to act like macrophages of the nervous system. They're going to help engulf foreign material, and they have that immune function, whereas astrocytes are going to be more of that support. So we've talked about the blood-brain barrier that is formed by astrocytes, and so they are going to help keep the cells within the central nervous system kind of separate from the rest of our body. And in addition to that, if we have some sort of injury or infection, inflammation, things like that, astrocytes can kind of move to wall off those damaged areas. So they have a really cool function with kind of the support side of things, whereas the microglia are more involved with phagocytosis and engulfing foreign material. All right, last up we have the spinal cord. So we have seen now the spinal cord cross section from a model perspective, but I could ask you this under a microscope. So let's go ahead and compare structures here. So first thing you always want to do is orient yourself. We need to find our ventral versus dorsal, or anterior versus posterior sides. And one big giveaway is we've got this nice dip here. Remember, the big fissure is going to be more anterior. So we're going to call this the anterior median fissure. Posteriorly, we don't have that extreme dip. So in this case, this is going to be our posterior median sulcus. A sulcus is going to be less dramatic than a fissure. We can also use the horns to help us. So we've got our lateral gray horns, anterior gray horns, and then the ventral or posterior gray horns are a little smaller, and they come to a point. They definitely look more horny. In the middle, we have our central canal, and connecting the two pieces of gray matter here, we have our gray commissure. So remember, gray matter in the spinal cord is going to be more interior, whereas white matter is more superficial, and this is reversed from what we saw in the brain. Next, we have our roots. So coming off of the horns, we have these dorsal and ventral roots, and we're also going to have a dorsal root ganglion. So you can still use your anterior versus posterior orientation to help you identify these, but another big giveaway we have is this enlargement here. This is our dorsal root ganglion, which is going to contain those neuron cell bodies. So this, we know, always has to come off of that dorsal root. So by identifying dorsal root ganglion, you can then find your dorsal root, and this leaves this other guy as our anterior or ventral root. Okay, so these are all things we saw in the model. They're all things that you can identify under a slide. However, sometimes our slides won't have this extra information. Sometimes all we will be able to see is this cross section. So when you're studying these, I would focus on all the structures you can see within this view. Don't worry as much about the roots under a microscope. And again, if you want to check your work, we can click Show Labels, and you can check your knowledge against these labels.